Our next session is patient-focused drug development in the age of cell and gene-based therapies. So happy to hand it over to our co-moderators today, Heidi Grabenstatter and Collin Hovinga from our rare and orphan disease programs.
Thank you, everyone, for attending the session. This will build on some of the other focus sessions talking about PFDD. We've had an opportunity over the last 2 days to have sessions dedicated to this. We wanted to give a little bit different topic for this session. Given the interest in regenerative medicines, it's a very different landscape from small molecules. Some of the questions are the same, others not so much. So what we thought we would do is we would assemble a team of really great experts to lend us some ideas on their perspective at different facets to regenerative medicines. And so we're going to start out this session with just introductions and where are people from, and then we'll dive into the individual questions we thought. And at the end, we'll open the forum to the audience so you can ask your own questions. So Lola, do you want to start?
Sure. I'm Lola Fashoyin-Aje. I am a medical oncologist by training. I am the director for the Office of Clinical Evaluation in the Office of Therapeutic Products at CBER, the FDA, and our office reviews all cell gene therapy products that come to the FDA, and it's a pleasure to be here.
Hi, everybody. I'm Steve Roberds. I'm the Chief Scientific Officer at the TSC Alliance, or a patient advocacy group based in Silver Spring, Maryland, serving tuberous sclerosis complex, a relatively rare disease, not ultra rare, but certainly an orphan disease that involves epilepsy, tumor growth, neuropsychiatric issues, so on, throughout a lifetime. I've been there 13 years. Before that, I had a 16-year career in the pharmaceutical industry and research.
Hi, everyone. I'm Ed Neilan. I'm a pediatrician and medical geneticist. My primary role now is as the Chief Medical and Scientific Officer at NORD, the National Organization for Rare Disorders. But I spent most of my career at Boston Children's Hospital and Harvard Medical School, where I also retain appointments and serve part-time. And I spent about 5 years working at Sanofi Genzyme.
Hi there, I'm Melissa Lestini. I'm with the FDA. I work in the Office of Pediatric Therapeutics, and that's actually located in the Office of the Commissioner. So as a neonatal consultant in the office, we help to promote and help with the considerations of all of the unique aspects of neonatal product development as those products come through the FDA.
Sonya.
Hello, I'm Sonya Eremenco. I am Executive Director of the Patient-Reported Outcome Consortium at CPATH. I've been with CPATH for just over 8 years, and I have a long history, over 25 years, in the basically what's now called the CUA development space. I'm glad to be here as well.
Thank you. So, Steve, I'll have the first question directed towards you. You've obviously worked a lot with pharma and with your nonprofit, and obviously the patient community that you work with is often approached by different companies in different states. Can you, relay some of your experience on like good practices and bad practices that you've seen, or things that need to be improved, and maybe some things that you communicate to the audience, since we have multiple stakeholders from different roles, and like what they should do in interacting with patients, particularly how it relates to consultations in regenerative medicines?
Yeah, so I think much like the early and often that we've heard from FDA, it's the same thing with patient advocacy groups. We have we not only serve the advocates, but we also have research programs. We have grants like many others, but we have a preclinical consortium where we've engaged industry with a set of models that are relevant to TSC. That gets us involved at a very early stage. I think it's important to make sure that the companies understand what all the organization, the patient advocacy, that the community can bring, because it can differ from community to community, the level of the number of patients, the level of how long they've been around, that sort of thing. But still everybody, but it's very similar things, right? Having that patient viewpoint. I think we'll talk more about some of those specifics.
Thank you. Ed, similar question from you. I mean, you've also had roles in both pharma and you have a really overarching role within NORD and are involved in a lot of different initiatives. I mean, can you share some of your good experiences and positive experiences engaging in PFDD as well as the people that work with you?
Sure. So NORD is one of several organizations that help more disease-specific patient organizations hold externally led PFDD meetings at the FDA. Also, since 2019, NORD's had a memorandum of understanding with the FDA around being one of the partner organizations to hold listening sessions with the FDA. Maybe as we get into it, I might say a little bit more about that, but thinking that most people are already familiar with those things, and we could find a few examples specific to cell or gene therapy, but I'm assuming most people are familiar with that. So I thought one of the things I would talk about is the newer Bespoke Gene Therapy Consortium. I won't presume that everybody knows about that. It's a public-private partnership coordinated by the Foundation for the NIH, with the main partners in it being NIH, FDA, and about 15 private founding organizations, as well as a few others that have joined. Most of the founding private organizations were, sorry, is that better necessary? Most of the founding private organizations in the Bespoke Gene Therapy Consortium were pharmaceutical companies, but NORD and Cure Duchenne and the Alliance for Regenerative Medicine were amongst the nonprofit organizations that joined, because obviously the diseases that we're concerned about, the rare and genetic diseases, you know, are the main targets for gene therapies. One role that I've had for the last about 2.5 years or so, representing NORD on that consortium, has been to be the co-chair of the clinical sub-team of the Bespoke Gene Therapy Consortium. And after the consortium launched publicly in October of 2021, we spent about 18 months running a competitive application sort of process to pick the diseases in the clinical trials. It was a 2-stage application process. Ultimately, we picked 8 diseases and 8 outlines of a clinical trial approach, and the scope of the consortium is to carry out first-in-human AAV gene therapies for 8 very rare genetic diseases. They were intentionally chosen to be rare enough that they weren't really subject to commercial competition, so that the dozen or so biopharmaceutical companies involved are able to collaborate without difficulty. And that's been a wonderful thing in and of itself. But now that we've selected the diseases, contracted with the clinical sites, and are looking back again at the clinical trial protocols and sharpening them up to actually, have some more INDs granted and start clinical trials in the next year or so, the foundation of the NIH led an interesting effort to sort of systematically pull in the patient perspective for all 8 of these gene therapies at once. I checked a couple numbers so I could speak to this with some authority. So 8 diseases, 10 different patient advocacy organizations, and in the initial workshop, about 15, 16 patient representatives. I think the main achievement so far has been to actually embed at least one representative of the patient advocacy group directly into the cross-functional team for each of the 8 diseases. And to some extent, the cross-functional teams in the Bespoke Gene Therapy Consortium are modeled after things that, like, I experienced at Genzyme, or other people from industry will have experienced, a cross-functional team with regulatory experts, clinical experts, preclinical experts, manufacturing input for each product, for each gene therapy that we're developing. Embedded right into the team is now a representative of the patient advocate. And they've begun reviewing the protocols. Now, there was already some patient input into the applications that we received from the academics who proposed that these were appropriate diseases, and we asked them for that input. But now that they've become Bespoke Gene Therapy Consortium projects, we're having to own that And I think it's interesting how it's been systematized that all 8 of the programs have this embedded representation. They started to have some real input into the clinical trial. So for example, pointing out for one of them that the number of lumbar punctures being considered either needed more justification or needed to be adjusted. And the end result is we kind of come to a compromise, fewer lumbar punctures, good enough data, less burden for patients.
Yeah. I mean, in those conversations, just added a little bit of my own curiosity. I mean, you know, regenerative medicines are so much different than small molecules. We all can appreciate that. I mean, how do patients deal with sort of the risk benefit considerations and weighing that in their decision? And how does that shape the trial in those discussions?
Yeah, it's a good question. So for these diseases, there's no effective therapy. So patients tend to be willing to accept a lot of risk to have their first ever treatment. That's a little bit different from another use case that I looked at a little bit again, preparing for this. One of the patient listening sessions that NORD helped to organize, which was I think back in 2018 or 19, right at the beginning of the FDA patient listening session programs, was about gene therapy for hemophilia. And of course, that was different because there were available therapies. And so I look back, I wasn't involved directly, but I look back at the summary of that gene therapy related. It was a, if I didn't say it clearly enough already, it was a listening session specifically about gene therapy for hemophilia. And a lot of that discussion was about exactly what you talked about the risk benefit of that, given that there were already available treatments.
I could,
Go ahead.
if
That would
you
be-
don't mind, I could touch a little bit.
When was time?
Not to cut off the rest of
No,
the panel, but
perfectly.
we had a patient-focused drug discovery, externally led meeting back, drug development meeting back in 2017. And at that time, I didn't really think gene therapy was on the horizon for TSC. It's a very multi-system complex disease. But I had a call with a new CSO of one of the gene therapy companies that's working with us in the preclinical space, to just kind of, she wanted to get to know some more about the disease. I said, well, we've got this voice of the patient report. She said, yeah, I already found it on your website and I've read it and I'm there. So even though we did it not thinking about gene therapy, the main point of that meeting was about the burden to the patients, the current therapies, how they're working and how they're not working. And that's been very impactful and still is applicable. I think what we're getting now are questions in terms of the gene replacement therapy companies are, so how do we, what do we need that would essentially allow a patient or a family member to say, I'll volunteer to be first, because as you said, there are therapies in place, they're just not good enough. And so really building one of the things that we talked to companies about is building a strong preclinical package that you can show to savvy, individuals and families, to say, here's why this is worth trying, or maybe worth trying, because we've got this package, we've got a lot of data, we understand the dose we need to go in at. So it's shifted the conversation a little bit.
So that brings in, I'm gonna just hone on you two guys for the presentation. I'm sorry, but you're picking off each other really well. But I want to bring up with you, Ed, is that, one of the things that came in our prep session was, this is a bit of a plug for the NORD curriculum that we worked with you on, but, Steve mentioned like savvy patients. What opportunities are available to train people so that they can be a little bit more educated to understand the drug development process, and that you're aware of, that you might, point people to that may be, interested in understanding more?
Yeah. One of those resources that's new and therefore, worthy of a call out, is a new 12-module extensive course on the drug development process that is aimed specifically at patients and their advocates, although it may also be useful to other stakeholders. Even some of those in the pharmaceutical industry may want to watch some of this. This curriculum was produced by NORD in partnership with both FDA and CPATH, with funding coming through the RDCA-DAP grant. And the initial idea for this curriculum and the way that it is entitled online, I just want to make sure I state it correctly so you all can find it most easily. So the official title is the Rare Disease Drug Development Series, What Patients and Advocates Need to Know. It's not really restricted in its content to rare diseases, but that's the way it's labeled online. And one can go to www.learn.rarediseases.org and click the button for View Courses. There on NORD's website, you'll see courses that support how to form a rare disease organization. You'll see courses on how a rare disease organization can help to make their disease more research ready. And then you'll see the single largest group of these lectures and resources being the new drug development course. And that begins with the foundations of research, talks about the roles of different stakeholders from FDA to industry in that process. And the basic course syllabus, if you would, was developed by FDA. And every part of the materials has been reviewed by FDA. And I think it's a great kind of unparalleled, resource for people to look at. And as I said, on that same www.learn.rarediseases.org website, you can find other courses that kind of come earlier in the process. Another thing I think I'll mention, because I know was also something we talked about a little bit in prep meetings and is of interest. One of the differences between, regenerative medicine therapies, gene therapies and the like, and the small molecules is that, if you're taking a small molecule and you stop taking it, it's out of your body in 6 hours, 8 hours, 72 hours, whatever the half-life determines. The gene therapies, you've made an irreversible intervention of some kind. And so the question of follow-up and the length of follow-up is a greater question. And so jumping back for a minute to the Bespoke Gene Therapy Consortium, last month we held a workshop, again, representatives from the stakeholders, NIH, I think there were some FDA people there, but mostly NIH and FNIH and the patient advocates involved in the 8 Bespoke Gene Therapy Consortium diseases. And we already started a discussion about the likely 5-year or longer follow-up and how maybe the patient advocacy organizations could help find ways to, explain the need for, lessen the burden of, and hopefully, sustain good, appropriate long-term follow-up for gene therapy.
Okay. I'm going to switch gears a little bit and switch to our FDA colleagues. And perhaps you can review so everybody is at the same table. I know we all assume that everyone understands the ways that we kind of interface with the FDA as patients and patient-focused drug development. Perhaps one of you can begin a discussion of what are the mechanisms by which people can use it, how they might differ, and then maybe some personal insight to how you have used their insights in your own decision making or thought processes as you begin, particularly how it relates to cell and gene-based therapies. Do either one of you want to start?
Go ahead. I think
I'm
during the
fine.
prep session you went first. So
I think that so in general, in terms of ways that the FDA hears the voice of the patient, there's certainly the FDA sponsored listening sessions for patient-focused drug development, and then also those externally led sessions. As part of the Office of Pediatric Therapeutics, there's a physician from our office that attends every pediatric-related listening session that occurs, and then reports back to everyone else in our office at our monthly meeting to debrief and discuss what the disease was, what the need is, what patients are saying, especially in terms of things that affect trial design, things that risk-benefit considerations. What are clinically meaningful outcomes that patients would like to see from these trials? And I think that there's a lot of value in that because, sitting in the office of the commissioner, we then can affect other offices as they come to us for help and can help to disseminate that information. And certainly we act as advocates in helping to ensure that those voices are heard more widely. I think the other area that we see this in is also a voice of the patient reports, and certainly, you know, when files come in, when they've done focus groups that focus on specific areas of the disease and various aspects in trial design, that's always incredibly helpful as well, and always something that we take into consideration.
Okay. Do you have anything to add, Lola?
Yes, I'm happy to. I think, you know, just to amplify some of what has been said already, there are, you know, formal mechanisms by which we regularly get the patient voice to help inform regulatory decisions and how we give advice to sponsors. You've heard already, we have a formal patient-focused drug development program where there are externally or FDA-led listening sessions. And those are really critical to the staff because, as you know, there are many, many rare diseases, and it is impossible to have an expert for each of those diseases on staff. And so really what we learn from patients regarding their experience with the disease and what outcomes are really important for them, and understanding what risks they're willing to take to be in a clinical study is really important and critical to our assessment of proposals that we review. There are obviously also, you know, patient-reported outcomes that are included in clinical studies that provide really valuable information, and that really give us sort of how the patient is experiencing the treatment, right? That in a way that's very different than when a physician is the one sort of reporting what the patient experience is on the study. There are also informal mechanisms by which we get feedback and input from the patient. And I think those tend to be sort of glossed over, but we really get a lot of rich information from those informal interactions. So examples are, you know, participation in workshops where we're discussing things around clinical trial design, outcomes that are important, even sort of how to design a study in a way that does not, you know, lead to a lot of burden for patients. Those types of meetings are really important, and they really frame our thinking as we apply the regulatory framework in giving advice to sponsors. There are lots of physicians in our group who are still taking care of patients. And there is, you know, I want to really, I can't overemphasize really what that experience provides in terms of really reviewing the products that are coming through, the proposals that are coming through, because you know you have that direct experience and connection to patients. You know, patients obviously also participate in our advisory committee meetings, either as special government employees on the committees or in part of the public, you know, comment period in these advisory committees where they're providing input on specific data that FDA is reviewing. We have opportunities to engage with, you know, organizations like NORD and others in really trying to customize the programs that we're reviewing to address the specific needs in a specific community or specific disease setting. So you know, I think there's not a situation, as I mentioned in our prep session, where the patient voice is not, you know, really, you know, critical to including and considering. And I think what is sometimes may not be satisfying, I guess, is that we don't do a great job at documenting how that patient voice impacted our decision making. But I want to assure you that it is front and center as we consider these therapies. Even when we're reviewing statistical analysis plans, you know, what treatment that is administered after the gene therapy and that has a potential to impact clinical outcomes. How do we handle that from an analysis standpoint to ensure that patients have access to a rescue therapy, as it takes time for the gene therapy to have its effect, but also so that we have a rigorous study where we can conclude at the end that the treatment was effective. So at every single stage in the design of the study, in the analysis of the study, in the interpretation of the study, the patient voice, whichever way we get it, through formal mechanisms or informal mechanisms, really has a significant impact in our work.
I want to, the panel before, one of the audience members asked about biomarkers and early biomarkers with the NICU babies. And I think patient-focused drug development is not a one-time thing. It's this iterative process, and that we learn as we go. And I wanted to highlight and ask Steve about the biomarker that they found early on, and where we draw this line in between small molecules and gene therapy. The other thing I want to steal from an earlier panel was the road testing, because I think there is an opportunity where you can road test the biomarkers in the small molecules, and they have a prognostic potential for later gene therapy trials. And also, and to talk about the education, an ability to inform your patient population, and that kind of circles back to risk benefit and decision making. So it all, I think everybody set us up very well, and to hear about your PREVENT trial and your biomarker, and it was not a gene therapy, but it set up a very nice framework for your group to move forward, and for your patients to understand a bit more about where they fall on the heterogeneity spectrum.
Yeah, happy to. About 10 years ago, or maybe 11 or 12 by now, a group of 5 clinics formed a consortium and worked closely with the TSC Alliance and they've had a number of NIH-funded studies, but one of the first ones was to basically test the hypothesis that one could see changes in the EEG in newborns with TSC followed at regular intervals with EEG prior to the onset of clinical seizures. So trying to get ahead of the disease. That was identified. Such a biomarker was identified, changes in EEG activity that weren't showing up as clinical seizures. And so that led to enabled the PREVENT trial, which was a preventative study to try to prevent the onset of seizures and ultimately developmental delay in infants with TSC. And so this was then the same group, but expanded to 13 sites, enrolled 86 infants with TSC, and using this biomarker then randomized them either to placebo or to vigabatrin, which is very effective at treating infantile spasms in TSC. So it really proved and the biomarker reproduced, it really has been road tested. The outcome of the study was the vigabatrin was very effective at treating infantile spasms in either groups, because if you went to placebo but started having infantile spasms, you would go on to vigabatrin. So it treated it very well and didn't demonstrate any difference in terms of development of seizure, drug-resistant seizures or developmental changes at 2 years of age, at least. So it was a little disappointing, but certainly the biomarker piece is still very valid, and I can tie that to gene therapy because, well, one of the observations in the PREVENT study was the best predictor of the poorer outcomes at 2 years of age was not which arm you were in, it was that the biomarker appeared prior to 7 months of age. So it's that early onset of EEG changes that in that study specifically, but it's also consistent with past studies, is very, that's predictive of the poor outcomes, the earlier the onset. And so when we think about gene therapy, we need to together figure out how to get it to those babies who are going to benefit as soon as possible. And this comes to the safety issue and letting FDA hear from the patients and the parents of these young kids who have had these poor developmental outcomes, and who might say, well, then we can ask them that, right? Okay, if you saw this change and you had these data from PREVENT and gene therapy was an option, you know, what are you thinking? What, you know, do you think? So we could get that sort of direct feedback.
See if I can hear me. There we go. Sonya, you know, we're gonna talk about other topics relative to regenerative medicines. You know, one of the things that we've touched a little bit on the panel discussion is that obviously the difference, particularly for long-term durational effect, right? And you know, the questions we always have are, you know, sustained benefit. What are the risks, and what does sustained benefit mean? You know, and you know, what are the monitoring for safety concerns? Obviously, you work a lot in the COA space and are very familiar with the roadmap, which everybody, thank you, Michelle, has been kindly educated on. But can you highlight how, you know, the patient voice can influence what things we should measure in that long-term follow-up, how that might impact what we actually look at and what the duration might be, even if I mean, that's a riskier question, but the things that might be important and how that might be collected, because my guess is that there's a lot of different opinions, but those may not always include exactly what the concepts that might be relevant to patient. But do you have any thoughts on, you know, what we need to do there and how that might be done?
Yes, thanks so much for that question. So, and I want to go back to partly what Dr. Fashoyin-Aje said about the importance of the patient voice and really wanting to know what's meaningful to patients, but also thinking about the context that Steve was talking about, which is a lot of these studies are probably going to be done in young children, and those young children probably can't report for themselves. So we need to think about other types of COAs. So when I think about patient-focused drug development, I'm thinking about the roadmap, I'm thinking about the guidance series, I'm thinking about all the COAs, and part of that roadmap, it was understanding the disease, it was understanding the benefit, the concepts of interest, the context of use, and it was also selecting the reporter, the best reporter for that context of use. So in some of these cases, it might need to be an observer-reported outcome measure, which is the caregiver, the parent, reporting on what they're observing with the child over time as the child grows to provide that insight into what's happening. But I want to, you know, come back to your main question, which was, you know, how do we understand what's important? And I think it goes back to guidance 1 in the series and guidance 2 in the series of the 4 guidance series, which is one is collecting comprehensive and representative input, and the other is identifying what's important to patients. And I think for a lot of cases where there's a lot of unknowns, like with gene therapy, you're probably going to need to do some of that foundational qualitative research, like open-ended concept elicitation research, to really ask the questions, ask open-ended questions, like what are their expectations, what's important to them. It might need to be longitudinal. That's something that we're starting to explore in the COA world, so to speak, is that we can do longitudinal qualitative research. It doesn't have to be a one-time cross-sectional, one time only. And for a case like this, I think it would make sense, because if you know, from what I'm understanding, and I'm new to the gene therapy world, I'll admit, you know, it's very small samples. I mean, these are very expensive treatments. They can't give them out, you know, they're not being given widely. So there's probably a very small number of people. So you need to really start with that qualitative research over time and collecting the information on what their experience is, what the side effects are, what you know, are they experiencing symptoms over time or not? Do these arise? You know, is it truly a cure? Is it really stopping the disease, or is it just delaying onset of symptoms? So, you know, to me, I think that's part of where the PFDD approach can really be beneficial, is to bring in those methods that are outlined in guidance 2 to really start to collect that information. And as you collect information and you start to see what we call saturation, you start to see the same thing coming up time and again, then you can start to develop a measure like a PRO measure or an observer reported outcome measure, or even, you know, could be clinician reported as well. That's one of the COAs to start to systematically collect it. And those can be a little more, you know, less burden. It's not as intensive as an interview, but, you know, perhaps that can be included in a registry. Then you're starting to collect data over time in a more quantitative fashion in a registry, and then you can come back to that and then start analyzing it over time.
Do you have like?
Yeah, I mean, I wanted to follow up on what both Steve and Sonya just mentioned, and I think, you know, we have a regulatory framework that allows us to try to mitigate the risks of a product. And I think that, you know, that's pretty established, and we work within that framework a lot of the time. I think in the context, especially in the context of rare diseases, it's also important to think about the risks of not having any options, or not having any options, you know, coming soon. And I think that that's where there's really an important need for customizing, you know, how we develop these programs, and just kind of going back to what Steve said, which is, you know, incorporating the risk tolerance in a particular disease setting when there aren't any other options. Understanding that, you know, patients, their caregivers, care partners are willing to take that risk. And so not being so rigid with the application of the regulatory framework, because it really has to be contextual. And I say this really in the setting of, you know, increasingly hearing from patients and patient advocacy groups around the need to really conduct these gene therapy studies before patients have had progressive deterioration and loss of function. And that's really something that we're constantly having conversations about within the agency.
Did you have anything that you wanted to add to that, or no? Well, I'm good.
I think you just threw out the context of the conversation, the parallels in the voice of the patient in terms of thinking about, you know, through my lens as a neonatologist, as a pediatrician, as a parent, as a regulator, what that voice brings. And I think that, you know, when I listen to many of these listening sessions and participate, you know, as a parent, I oftentimes find myself crying because they're rare diseases that have terrible outcomes. And I mean, granted, I'm focused in the pediatric space, so there's a lot of, you know, when I think about gene therapies, it's usually for something that's not good. And that then motivates me from my pediatrician lens and advocacy lens to get all fired up and want to do something about it. And then I put on my regulatory hat and say, how can we help this condition? How can we make sure that these patients' voices are heard, and making sure that what they have to say is incorporated into the study design, the outcomes, et cetera. And I think to what Sonya was saying, you know, in the pediatric space, and especially in neonatology, it's the parents that are making the decisions, and they're the ones that are really, I mean, you do have exact, you know, developmental outcomes and things like that that you can test, but there's some more granular aspects of the disease and how it affects the family and the family life that you don't always get at. And sometimes those things are very clinically meaningful to both the patient and the family. And I think understanding that and hearing the patient voice in regard to that is really important. And I just want to touch on another aspect of what Sonya was mentioning in terms of longitudinal follow-up and the importance of that. You know, I think, and it just triggered this thought in my mind when I was training in neonatology many years ago, the late preterm, those babies that are born between 34 and 37 weeks, they were kind of not considered a real preterm baby. And, you know, they'll be fine. Like they're not your micropreemies that are going to be in the NICU for 3 months or more. And through longitudinal studies, we've realized that, you know, these kids, although they seemingly are fine when they leave the NICU, can have some significant, you know, learning and cognitive delays at school age that are really impactful to the families. And I think that's also a parallel to thinking about the longitudinal aspects of following patients who have had regenerative therapies like gene therapies, and to truly understand, you know, the effects of the therapy, both in the short term from what the therapy, the, you know, potential adverse effects of the whatever vector and mode is used, or conditioning for the gene therapy, but then also those long-term effects of, you know, is this a lasting gene therapy? How does it, you know, is it something that works and it doesn't work, or are there, you know, longer term issues that we just don't understand? And unfortunately, especially when it comes to pediatric rare disease, it's not something that can always be modeled easily. And oftentimes we're relying on a lot of non-clinical data to help support that first in human study in patients.
I'm interested in, given the spirit of innovation, and I want to maybe ask the audience too, but given Sonya's expertise, some of the patient-focused drug development sessions are recorded.
Mhm.
And it was great to hear that what Steve said, that the voice of the patient document that was built by the TSC Alliance right away was sourced by a pharma company. But some of the most interesting insights are what you hear from the audience members related to what Melissa was saying about the caregiver burden and some of those other sort of qualitative things that you would want to measure, and how you might do that in a neutral, collaborative convening. How could we look at those videos and those recordings that have already been done of a number of different disorders, and what survey, what COA methodologists often do is like the talk out loud inside meetings, right? And there's technology to kind of look at that, right? And that's data.
So I guess I want to qualify that. I would call that's patient experience, like it's information.
Right.
I would hesitate, I'll be honest, I'll hesitate to call that data the same way we consider like the qualitative interviews data, because those interviews are being done. And this goes back to the definition of PFDD, which is systematic approach to collecting patient experience data. So, you know, we want to have, for in my view, which might be considered a little strict, but for it to be data, we need to have a protocol. We need to have an interview guide. We need to have like more or less asking the same questions to each participant and having those questions be asked in a neutral way, not a non-leading way. Because, you know, there are, you know, risks with if you ask a question a certain way and it's leading and you get the answer you're looking for, but that's not truly what the patient's experiencing. So, you know, so I think to me, the listening sessions and the PFDD meetings provide a lot of really good information about the experience with the disease, with the treatment, with what's not, you know, what the frustrations are, what's not available, what they would like to see. I've attended some, I've heard some, and those experience, the voice of the patient reports, I think really do serve that purpose of describing the context, describing what's really happening. But to turn that into data, I think you really need to have something more systematic. And so those, I would see those being used for is to help you design your interview guide, to really identify, okay, what are the key concepts of interest that I want to make sure I talk about? Because the other challenge with qualitative research, especially when it's open-ended concept elicitation, is you're going to probe on certain things, but then you want to have it open enough to get things that you didn't expect. So you need to have that flexibility in those interviews, but you still need to have an idea of, okay, I'm expecting to hear certain things, and if I don't hear about that, let me go back and probe and clarify if that's really happening. And I can give you a quick example. I know it's not a rare disease, but it's a quick example of how this is important. So we've been working, a group of us at CPAT have been working with colleagues at Northwestern on a secondary analysis of interviews with Parkinson's disease patients. And what ended up happening is, like, initially those interviews were done, I mean, they're done very well, very thoroughly, and patients would be asked, okay, what are your symptoms? When were you diagnosed? What was happening when you were diagnosed? And they would list all these symptoms, and then they didn't mention certain things that they didn't associate with Parkinson's. So they didn't mention constipation. They didn't mention depression. They didn't mention things. And what most of us would say, well, is that really, that's part of Parkinson's? And it truly is. Later interviews, the interviewers realized this was happening. They would go back and ask the patient about those. And so we have sort of this gap in our data where we have certain concepts that weren't endorsed, quote unquote, but it could have been because the patient didn't recognize it as a symptom of Parkinson's. So I think that's where having that voice of the patient information, where we at least have an idea of, okay, here's what we should be hearing about. And if we're not hearing about it, let's, we have to probe further. I don't know, does that answer that? I'm getting a little more into the weeds methodology.
Well, like a follow-up question, I think, because like how I think about this is like, the conversation that you just were mentioning, I think if I were a family member asked those questions, I wouldn't necessarily think about it in the context of something that was permanent, right? I mean, I might understand about my IDs, but if I was talking about in the context of oh, you're going to give me a treatment that alters my genome and it's on board for the rest of my life, and how I determine how I feel about this or that or the other thing, and maybe you're different. Are there anything in these discussions that that have come up in either focus sessions that the agency has had, or even the experiences that you've had that are similar to talk about, or the late effects, or how you might weight things differently based on the type of treatment, particularly the risk benefit determinations? Hopefully I'm clear with that. I mean, because if you're asking about disease and the concepts, the lack of weighting and the risk benefit calculation, those discussions, yeah, it's like, oh yeah, I had constipation, but if it cured me, would the constipation be something I would weight differently? And I don't know if that's teased out in those methodologies or whatnot.
I would say probably not yet,
Yeah.
because it's just such a new area.
Yeah.
But I think it's something that really needs to be thought about and considered
Yeah.
as we move forward, as we continue to move into gene therapy.
Do you have other questions?
I think it'd be great.
Yeah,
to
we
hear
can
from the
switch to the
audience.
audience.
The
Okay.
audience has questions.
This topic came up yesterday during the one session about missing this. And I think we're entering that conversation again. And I see this in the translational science space, Micah over there, there's this valley of death we always talk about. But when we're talking about cell and gene therapies, these regenerative meds, I think we're facing a new valley of death where these companies abandon studies, or, after early phase and these patients have been treated and this long-term follow-up, the guidance says contact the FDA for what to do. And I wonder how many companies are contacting the FDA and what happens to these patients and all this data from a therapy that has altered their genome potentially, and they fall in this valley of death where maybe even the physician who followed them isn't their primary care physician. So their primary team doesn't even know what to report, how to report, what to look for. So I wonder if there's any comment to that in this new valley of death and how we address it.
Want to have any response?
Yeah.
Any other questions from the audience? Yes.
Thank you. Admittedly, I haven't read the guidance documents other than the titles. I just looked up on my phone, but I did want to ask, is there guidance on translating a patient need for improvement into something that fits a more clinical scale? Like our patients don't think of improving their SARA ataxia score. They think in terms of being able to use the bathroom by themselves or to get into the car or something like that. Is there guidance on how to take that kind of patient-derived need and put it into something that's more clinically quantifiable for evaluation?
So I would say it's a combination of guidance 2 and guidance 3, because guidance 2 is really about identifying what's important to patients. And then guidance 3 is a little more about how we turn that into a measurement tool. Because that's, I think, what you're asking, is how do we take what patients are telling us and then we actually turn it into a measurement tool where we can actually quantify the results. So, and I don't know if you were here when Michelle spoke about the PFDD guidance series. I think it was yesterday, where she said, I know it's a lot, it's scary, it's overwhelming. And it's true, guidance 3 is kind of overwhelming in that way, but that's really where it goes into, we talk about the concepts of interest, we talk about context of use, and it really does dive into that. But it is a very challenging process. It's not easy. I will not water it down. It's a challenging process. It takes skill, it takes understanding. It takes people who've been working in the field to really do that translation, because you're right, there is a translation. I know, and when it comes to PRO measures, we actually do try to use the patient language to the extent possible. And when we're developing a PRO measure, so we take that from the qualitative interview data, but sometimes it just doesn't quite work. Sometimes it's just either too colloquial or too, too vague. So we do have to try to make it into something that we can measure.
In my previous life, working with patient advocacy, I worked with a clinical research network that they were doing the opposite direction. So they were they were developing COAs and validating them across the centers. And then once they had a validated COA for a certain domain, they were making the patient reported or caregiver reported outcomes for the validated COA. And they were still using the FDA guidances that you alluded to. But it was, if I understood your question correctly, you were talking about going the opposite direction and starting from the patient reported and then making a clinical outcome assessment.
Hi, this is Micah Averts. I'm at CPATH as well. I have a question not about the process, but about a specific question related to gene therapy and especially viral-based delivery factors. With the whole concept of re-administration is typically not possible. So what are some of the patient concerns or thoughts around if I enroll, especially a child, right now in this trial, I'm pretty much excluding him or her for any future similar vector-based therapies. So I've heard about this concept in the abstract, but I just wondered if there's actually some active discussions around it and what the feelings and thoughts are of the patient community around this.
Well, I can say we haven't solved that, but it's an active topic of conversation to understand and dial in that risk and how people will react to that. Because that's kind of part of the reason for needing just as strong a package as possible, because we are thinking about it as being AAV based, as being one and done from that perspective. And so what's it going to look like? What is the follow-up going to be? Are they going to look at TSC, which is multi-system, as a holistic disease, not just the seizure counts, right? But what matters to patients, but understanding, we're still in the midst of understanding that, but it's an important topic. Yes.
Any others want to respond? Okay. Any other questions in the audience? Lynn? Anna? Yeah.
So just to provide a follow-up to that, Lynn Yao, FDA. And this goes to my time as a clinician, actually. It reminded me when you brought up the question, I asked a patient once because, and this is kind of how I thought about things as a clinician. As a pediatric nephrologist, we would have patients on dialysis, and then the opportunity might come for transplant. And, you know, transplants don't last forever, at least and they still don't, but certainly at the time that, you know, I was actively practicing. And so we used to kind of joke in our field that we were just changing ICD-9 codes, right? It wasn't really like you were curing anybody with a kidney transplant. But I asked a patient and I said, look, does this really make a difference that instead of dialysis, you know, with all the potential complications and that, you know, you have all the risks of a kidney transplant after you have the transplant, is this really better than dialysis? I asked the mom of a patient and she said, and the one patient said, I would take one day as a transplant than every day of my life hooked up to that machine. And the mother said, Lynn, you don't understand, because when my son is hooked up to the dialysis machine in the crib at night, you know, they can't run into the room and when they wake up in the morning and give me a hug. I have to go in and I have to unhook them. So my point is that even in these hard discussions about, you know, what does it mean long-term follow-up? What does it mean? You know, how do we collect these data points? That the patient voice is so important in figuring out for any individual patient, but also in terms of what we're going to do in terms of collecting long-term follow-up.
Thank you. Tom?
Tom Miller, Bear. Maybe more of a comment than a question, and maybe a bit more specifically relevant to sponsors that are thinking about rare disease studies, pediatric or otherwise. But at least in my experience, we're seldomly able to execute a study in a single country. And because of the scarcity of certain rare diseases, we may need global cooperation to be able to effectuate a study with feasibility. And what we've learned, what I've certainly learned through the years, is that the patient voice and the patient perspective may be very different depending upon which country you're asking it in. So I think it's very important, certainly for sponsors, but I think for all of us in our ecosystem here, to recognize that perspective in the US may be very different elsewhere in the world. And if we hadn't, in the instance that prompted me to make this comment, as that I was recalling, made a deliberate effort to try to understand that, there would have been parts of the world where we would not have been able to enroll studies. So would encourage everyone in the ecosystem to think a bit more globally.
Well, I think that's we're close to time. Thank our panelists for your participation on the call, the section. This is exciting and it's a different time.